Good morning, one and all present here. Kindly confirm my audibility.
Yes, ma'am.
Okay, thank you. Now I request Ms. Krishma, Assistant Professor at our department, uh, to deliver a welcome address. Ma'am, please.
Yeah, thank you, ma'am. And is my voice clear?
Yes, ma'am, clear.
Yeah. So, good morning, everyone. And on behalf of Department of Microbiology, Ratnam College of Arts and Science, I extend a warm welcome to all of you at this second day of a two-day national level lecture workshop on recent trends in life sciences amidst a COVID-19 pandemic and insight into innovation. And I find immense pleasure to welcome our chief guest, Dr. Mridul Umesh, Assistant Professor, Department of Life Sciences, Christ University, Bangalore. And we are really thankful to him for taking the time out of his busy schedule. Welcome, sir. And I feel honored to welcome all the delegates of this workshop from all over the country. And I would welcome all our participants and I invite you all to participate and present your views on this topic. And I extend a warm welcome to all my fellow colleagues for all your support in this important program. And I welcome you all once again to this workshop and I hope you all will have a great time ahead. So thank you all.
Thank you, ma'am. It's my pleasure for having an opportunity to introduce our guest speaker, Dr. Mridul Umesh, Assistant Professor, Department of Life Science at Christ University, Bangalore. To start with, he was a guest faculty and assistant professor at Guruji Institute of Science at Coimbatore. He is an expert in bioprocessing technology, bioinformatics, and clinical microbiology. So far, he has published 30 articles in reputed peer-reviewed national and international journals. To his record, he has received notable awards like Proficiency Award at SNGC, Coimbatore. Till now, he has delivered four wonderful lectures at various institutions. So, once again, welcome you, sir. Now, the session over to you, sir.
Thank you, ma'am. Am I audible?
Yes, sir, you are audible,
Okay.
sir.
I'm just taking one moment to share my screen.
Sure.
Uh, Hope it is visible to you, right?
Yes, sir, visible.
Okay. So thank you so much for the opportunity and a good introduction. I'm just privileged to deliver guest talk on this particular topic, that is integrating waste valorization for biopolymer production, sustainable approach. So before we started, just like all online classes, what I used to tell to my students, in case if there is some technical glitches in between, I don't think it will be there, but in case if I'm facing some issues, I'll just connect back in another 2 minutes. In case if it is there, so kindly just wait in case my connection is lost in between. Okay. Yes. In this interesting morning, what I wish to deliver to you is something about our sustainable approach and why we have to work on biopolymers and how we have to connect our waste management strategies with the biopolymer production. So before I start with my lecture, I would like to tell to you, like I'm working in multiple, biopolymers basically. Some of them you would have heard, and some of them would be totally new to you. But I just want you to focus on different types of biopolymers, and my prime interest was on microbial biopolymer. It is basically polyhydroxyalkanates. You would have Some of you would have heard about that particular thing, PHA. We call it generally as PHA. So that was my research area for my doctorate. After that, I was concentrating on many other biopolymers, and everything what I work is basically emphasizing the concept of waste to a product formation. Okay, so I'm just going to give you some of the research designs or how to frame a project, so that what I thought is, rather than explaining the hi-fi things regarding this is this and that and in abroad whatever is happening, I restricted all those things, like what I did and what you can do also in your college level, maybe for an MSc project or maybe for a PhD project at whatever facilities, because I basically, my education was also from Coimbatore, everything was from Coimbatore. So I know that what are the facilities, what would be available to you, what would be the access, what would be the limitation also. I mean, we cannot think about, every time we cannot actually think about the standard of IIT and IICs and all those reputed institutes in a small college also. But with whatever minimum facilities you have, still you can work on this particular thing. So that is what I would like to tell you, and that is how our presentation goes. So let me begin with the interesting chord. Why we are not concerned about the safety of our environment? So because our trees doesn't provide us with the Wi-Fi signals, right? So once one day, just imagine the situation, especially during the pandemic days, uh, we are very much troubled if our network is not actually working, if our Wi-Fi is not actually working, right? So at least for 1 minute or 5 minutes, we'll be getting tense. Like you can survive without food and water, but without mobile, without Wi-Fi, it is difficult these days because you'll be getting out of comfortability zones once your network or your range issues are there. Wi-Fi is not working, the net is not working. This particular things, but why we are not having that kind of a curiosity in conserving our environment? That is the prime question. Now, let us begin by examining ourselves with a question. Is this the time to think about all those things? Like we talk a lot about environmental conservation, right? We'll say like tree cutting is should be stopped, like soil conservation has to be there, microbiota conservation has to be there, plastic should not be used. But all these things are our strategies. But do you think this is actually the time to put on these strategies, or do you think like this is the time to act? Okay, so the next pictures will clearly explain to you what I actually want to say. Okay, or why bioplastic or why biopolymers as such, is it the time to think or the time to act? So I don't want to explain anything because the picture itself will give you a clear-cut idea regarding what I actually want to tell you. Right? See, who is the inventors of all these things? Plastic, no doubt, it is the wonderful invention of mankind. I have no objection with that particular thing because you cannot find a material whose properties or whose characteristics are such what to say? I don't get my words in my dictionary to refer to that particular. It has an excellent properties, wherein it is touching every sphere of your life, right? Right from the mobile that you're touching, it has some kind of plastic parts also somewhere or the other. Where the water bottle that you take, from every sphere, from just think from the morning to evening, whatever you use, the use of plastic is inevitable. But it is we who has devised this particular thing, and it is we who are utilizing this, and finally who is actually being affected the most. See, these creatures, they are in no way responsible for either their creation or either their disposal. But finally, those who are into trouble is these particular creatures. And thereby, we have a commitment. We call ourselves as a super creatures of the nature, right? So we have our own commitment so as to protect the integrity of the environment and to conserve the integrity of this wildlife. Otherwise, what happens, no, like how we are looking at into the tiger and lion these days, maybe after 50 days, we should show the pictures to our next generation and show that I have seen this particular thing, but it is no more existing. So that will be the story. It's not just a like a, what to say, extrapolation or something like that. This is what is happening. There's a lot of species which is being affected by something called as microplastic pollution. Microplastics are the most dreadful thing which is actually increasing nowadays. You cannot see them, but they are actually accumulating inside the system, even in humans, fishes, and all those things, and it is causing a great problem. So I'm not touching those spheres. So all these particular things emphasizes the requirement of the bioplastics, or the usage of biopolymers or bioplastics, which is basically degradable in nature, and how you can design a work on this particular thing. So biodegradable polymers. So I have mentioned synthetic polymers over here. Why? Because there are some kind of synthetic polymers which you can create, and it has a good degree of, still has a good degree of biodegradability. I'm not telling about 100% biodegradability. Still, it is supposed to have a good degree of degradability, and thereby you can actually use them for your further applications. Okay. So the next one is natural polymers. I think somebody's mic is on. Can you just mute? Thank you so much. Yeah. So the next one is natural polymers. So what about natural polymers is these are the thing which is present all over us, and my target is basically on this natural polymers. And I'm working with chitosan, and I'm working with chitosan. I'm working with starch. I'm working with dextran, and in protein-based polymers, we are working with collagen. Say we are actually starting for working. Apart from that, then we are working with hydroxyapatite, and we have started with cellulose. Few papers with cellulose also. But the major theme of the presentation is all about polyhydroxyalkanates, which are bacterial biopolymers. The biopolymers which is exclusively synthesized by this bacteria. Okay, so I'll give you a total insight on how to work with polyhydroxyalkanates, which is one of the promising biopolymer, and it has got right from it's almost 100 years since it has been discovered, and still it has got a high commercial value, and there's a lot of research which is happening all over the world, and you have in a year around 40 high impact factor papers which is published on research pertaining to polyhydroxyalkanates. So what are these polyhydroxyalkanates? As I told you, this is a class of biopolymer, and in chemistry these are the polyesters of your hydroxyalkanic acids, HAs. So this is the structure, and they are synthesized by numerous bacteria as intracellular carbon and storage compounds. As you know that there could be some kind of inclusion bodies in your basic microbiology. You'll be studying inclusion bodies, whatever is present inside the bacteria, and what is the role of this inclusion bodies. Once the bacteria is under starvation, they derive the kind of nutrition from digesting this inclusion bodies. The same concept is over here. PHAs are a kind of inclusions which is present inside, which is formed inside the bacteria, and bacteria uses it for deriving its nutrition under stressed conditions. Okay, and the advantages is they are actually non-polluting. They do not need any catalyst for their degradation, and they don't want any additives to promote their degradation as well. So this is the only class of biopolymers which is supposed to be 100%, because we never have the concept of 100% degradation. It is around 99.9, but this is found to have 100% degradation. And a little bit about the history, like this is our tradition, right? Whenever we are actually studying something, we used to say like who was the first person to invent that particular thing in honor of the person. So this is what the picture first you could see is the inclusions of PHA, which is present inside bacteria. So this was first discovered in 1926. That's what I'm telling you. It's around, again, we are reaching 100th year soon. Still, it has got a lot of commercial importance, and it is discovered by a person called as Maurice Limoges while he is working with the inclusion bodies, study of inclusion bodies within the bacteria as such. Okay. Now, what is their advantages? Why we are actually so much concerned about this particular thing? Why it has got this much importance more than any other biopolymer which is present? It is all because this is exclusively produced by microorganisms as carbon and energy reserve molecules under nutrient limitation. So what do you mean by nutrient limitation is you generally have a huge amount of carbon and nitrogen in a media which is used to cultivate the bacteria, right? So once we are actually limiting any of the essential nutrients which is required for the growth of the bacteria, this bacteria will be a little bit stressed. So it actually channels its glycolysis pathway. I hope that you know the glycolysis pathway from glucose to pyruvic acid. So that is a general pathway for the production of ATP. So it will be deviating its glycolytic pathway so that it doesn't continues with the TCA cycle, rather it goes with another cycle whereby this PHA is actually synthesized. I'll tell you about this in the upcoming slides, how to work with that. So the advantages of this particular thing is under aerobic condition, if you see how will it degrade, it will produce carbon dioxide, water. So both are generally non-toxic or less toxic, or even though it's carbon dioxide. Anaerobic conditions, we have got a great importance because once pH is degraded, you will get the methane, which can be used for biofuel. So now the world is actually designing the works in such a way that we go for integrated production strategies for the production of this particular polymers as well as your biofuels. Okay. The advantages is these are biocompatible. It has a lot of application that I'll show you in one of the slide. What are the different ways in which it could be produced? But the major problem with the production or the commercialization of this polymer is its cost of production once you use a synthetic media. Problem is once you use a synthetic media, see, we are going to produce in like in gallons or in big fermenters. So you have to pay for glucose, you have to pay for nitrogen like ammonium sulfate or whatever the nitrogen source peptone you're using, and you know the cost of each of the things. You would have seen the bottles in your laboratories, right? So the current strategy is using a waste which is otherwise disposed into the environment and use that as a substrate for the production of this commercially important biopolymers. So that is what the concept actually emphasizes. So in that regard, what I'm going to tell you is how you can design a work based upon this particular things. Before that, these are actually the types of different types of pH. pH has different types, and the most studied type is the second one, that is the polyhydroxy butyrate. So this is a little bit about the chemistry kind of things. Means most of your microbiology students are not explaining about the side chain substitutions and all those things. We'll skip that. So this is actually the biosynthesis pathway. Although it is a bit, I mean, basically microbiology students, because my background is also pure microbiology. So basically, people from microbiology backgrounds, they are not that much, confident with the pathways. That's what I felt over these years, because here I'm seeing biochemistry background students as well as students. So again, but you should understand how this compound is formed. So whatever be the source of your sugar, it will be producing your acetyl coenzyme A at the end of your glycolytic cycle, so which usually goes to the Krebs cycle. Okay, but in case of your PHA producing organism, and once it is under nutrient stressed conditions, acetyl coenzyme A will be converted into acetoacetyl coenzyme A. I'm not talking about the enzyme mechanism and all those things. And this acetyl coenzyme A is then converted into hydroxybutyryl coenzyme A, which is the monomer of your PHB or your polyhydroxybutyrate, which is the chief class of your PHA. Okay, it's PHB is the most well-studied class, and this will be getting polymerized. You know what is polymerization, right? A monomer will be linked together, just like this beads in a string, and it results in the formation of the PHB. So this is the general biosynthesis pathway. How from sugar you could produce your PHA. Okay, keep this thing in mind because this is required once you go for the next line. Okay, designing the research work on PHA. So this is what I actually want to emphasize you. So how will I design a particular work on this PHA? First thing is we have to isolate the PHA producing organism. And what could be the sources of PHA producing organism? Anywhere you can. I cannot mention because if you search the literature, anywhere you can get this particular thing. You can take either water sample, sewage sample, you can take from garden soil, you can take from agricultural soil, you can take from way, you can take from whatever distillery effluent, whatever it may be. And you have to, there are certain medias, specific PHA production medias in the literature, where you can actually grow this particular thing. As usual, you see serial dilution followed by your plating, and you have specific staining methods called as your Nile red staining or Nile blue staining, which is a fluorescent staining method. So after you're growing this particular organisms in PHA production medium, that is agar medium, you have to expose this thing to UV light. And this media is incorporated with fluorescent dye, that is your Nile red or Nile blue. So the PHA producing colonies will be fluorescing, whereas the other colonies will be light translucent. The other wonderful methodology, but it is a little bit costly, and we cannot always afford this particular thing for the preliminary studies, that is a TEM imaging, so transmission electron microscopic imaging of your culture which is grown in the PHA production medium. But you can see like this clear inclusions as you could see inside this particular bacteria. So likewise you can clearly observe. So the first one is actually the isolation and screening of PHA producing microorganisms. And next one, the most common two methods, and it is relatively cheap. The basic chemicals which is required for doing this thing is relatively cheap. One is the Sudan black staining. So in the Sudan black staining, it's a simple protocol, just like a gram staining procedure, where you have two stains. First is the Sudan black, and the next one is your saffron, which is the counter stain. So this Sudan black is a stain which is specifically going and binding with your PHA molecules, and it will be seen as, you could see in the picture, like black color, whereas the cell appears to be a pink in color. So pink colored cells with the particular black colored inclusions represents the PHA producing organism. Next one is your acridine orange staining. Acridine orange is again a fluorescent stain. The problem is the stain is not that much costly, but the thing is that you require a fluorescent microscopy to study this particular staining methodology. So it should expose to fluorescent light, that will be fluorescing. The cultures which will be producing the PHA will be fluorescing. So again, these are the methods to check whether your isolate is having the PHA production capability or not. So the next one is, of course, there are not necessarily, you can target the PHC. So PHC is the gene which is present in almost all the PHA producing microorganism, because it is the one which is producing the enzyme PHA synthase, which is responsible for the polymerization reaction. Okay, so you can have a specific marker PHC which is specific for this particular gene, and it can be compared with your genomic DNA, what you could isolate from the organism. So molecular method can also be employed for finding out whether this organism is a PHA producing strain or not. So in that way also you can do this particular thing. So next one is what I actually want to target you, how integrating your waste for the production of your biopolymers. Okay, see what are the two different types of, I'm not talking about the protein part, because protein rich waste has a lot of applications. So that may not be used for the production of this particular thing, but I'm just going to talk about two major things. One is carbohydrate rich waste, and the other one is lipid rich waste, because most of our waste will fall onto these three categories: either carbohydrate rich, protein rich, or your lipid rich, which is the fatty acid rich. So protein rich does has a lot of application, so I'm not talking about that particular thing, but how you can use a carbohydrate rich waste as well as a lipid rich waste. So what could be some of the carbohydrate rich waste? Could be sugarcane bagasse, it can be fruit peels, it could be your distillery effluent, it could be your whey that is having high amount of lactose. A lot, if you sit and think, you will get a lot of waste in and around, food waste in and around us, which is having appreciable amount of sugars. Okay, this sugars, through glycolysis, it will be producing pyruvate, and once it is inoculated with a PHA producing organism, so that you should make this sugar in such a way that it is fermentable for the bacteria. So that is your task. Okay, so once the bacteria is fermenting, the PHA producing bacteria is fermenting that particular thing, you can generally, this cycle operate in them, and you will get the PHB molecule. So it is as simple as that. I'll get you with an example, a wonderful example. So this is a cycle. Once you use the sugar rich waste as a precursor for the formation of your PHA. which is biopolymer. Then lipid. So this is one of the area which is blooming as such. Okay, why? Because lipid-rich wastes are it's a great problem for the disposal rather than carbohydrate waste, because it takes time for its degradation in the environment, and it is so offensive, so that it produces a lot of putrefaction or the offensive smell. So generally, whatever is rich in fatty acids will be converted into acyl coenzyme A, and there is a fatty acid biosynthesis pathway that you would have studied in your biochemistry chapter, and then through this particular way, this will be producing PHA. Same way, from glycolysis also, the Linovo pathway, the fatty acid, biosynthesis pathway. So here it is the oxidation pathway, and here it is the biosynthesis pathway. Both will be channeled together. You need not do anything. Okay. So if a particular organism is capable of producing this particular PHA, it will be utilizing either lipid waste, or it will be utilizing the sugar waste so as to produce this PHA, because they are multipotent. They have the enzyme mechanism which makes them multipotent to produce from this. So lipid waste includes collagen, your chicken, the slaughterhouse waste, fish waste, many other things, lignocellulosic waste, which may contains which have a high oil content basically, or oil seeds, the waste of the oil seeds after extracting the oil, whatever you're discarding. So those kind of oil rich thing also could be exploited for the production of your polyhydroxy alkanes or your PHA. Okay. Yes, so I would like to give you some of these are this is not my papers basically, so I'll show you my papers at the end. So these are some of the interesting papers as per the literature where it deals with, see, this waste chicken feathers. So these chicken feathers, it has a huge amount of proteins, keratin protein is there. So even protein rich, I was telling you about the other things. So this protein rich waste, once you hydrolyze it, once you actually hydrolyze it with a particular bacteria, that can be used. So keratinase producing bacteria, enzyme producing bacteria can degrade them into simple fragments of amino acids, and using this protein, the PHA producing bacteria itself can produce the PHA. So what does it mean? It is not only carbohydrate, it is not only lipids. So the recent evidence has shown that even the protein rich waste could be exploited as one of the simple waste material for producing the PHA. So almost all kind of waste can be utilized for this particular thing. So again, as I told you, slaughterhouse waste. So what is the nature of slaughterhouse waste? Slaughterhouse waste is rich in lipids. So you have to target on a bacteria which is capable of utilizing the lipid as well as producing the PHA. So basically, lipolytic bacteria could be good examples, or the one which is capable of producing lipase would be good example, and they can accumulate. And the two things are, this should be lipase producing as well as they should have the capability of producing PHA. There's a lot of bacteria in the literature which can do so, and this is one of the way in which you can utilize the slaughterhouse waste, which is an offensive waste, for the production of your PHA. Now, another one, if you are interested in some non-edible oil or after extracting of the oil, because in South India, we generally use different kinds of oil like groundnut oil, mustard oil, coconut oil, gingerly oil, all those things. So after extracting these oils, whatever is left out, the residue, that whatever is left out can also, because it's still substantial, it still contains a substantial amount of your lipids. So this lipids could be extracted out, and that lipid can also be used as one of the ingredient for the preparation of this PHA, or through lipolytic organisms, by employing a lipolytic organisms. And second thing is, the after extraction of your biodiesel, you know that biodiesel, for the extraction you require the lipid part, then you go for your transesterification process to produce a biodiesel. So after that particular thing, there's a huge amount of waste which is left out. That particular thing can be used as a feedstock for the production of your PHA. So that can also be used for the production of PHA, and the cycle, the biosynthetic cycle, remains the same, how it is being produced. Then this is again another interesting one. This is not actually the waste, by the way, but I would like to show you this one also. One is apple pulp. So this is actually an interesting study where apple pulp is used as one of the instead of using the synthetic medium, apple pulp is utilized as one of the medium for the production of this particular PHA, which is actually obtained as a film. But you may have a question, like in a country where apple is costing around 200 to 300 rupees per kg, how will you utilize? So again, what I would like to tell you is this is not from India, it's from a study which is conducted outside. So how will you modify in your place? You know that in our country, a huge amount of banana is wasted. If you see to market, whatever is not sale for a week, it is getting spoiled as such, which has an appreciable amount of carbohydrates. Why don't you utilize that thing instead of apple? Because again, the concept is whether it is apple, whether it is banana, all we require is the sugars, right? So you can utilize that. You can modify the methodology and you can utilize it for the production depending upon whatever is available to you. So accordingly, you can modify and then you can have the process integration with the waste. So the basic step, as I told you on a summary, is hydrolysis. Hydrolysis, what is the requirement of hydrolyzing? Because this bacteria is a prokaryotic organisms, right? So they may not be able to take all the sugars. So if they are saccharolytic, it is okay. But if they are not saccharolytic, but if they are PHA producing also, then also you can do an acid hydrolysis, and the prepared acid hydrolyzed media can be used for the production of your PHA. If the bacteria is lipolytic, well and good, it can directly collaborate with the production of this one. Otherwise, what you do, you use produce lipase enzyme, use that lipase enzyme for degrading this particular thing, and the small molecules of fatty acids could be utilized by the other bacteria for the synthesis of PHA. Accordingly, you can have a process design depending upon the nature of your bacteria, depending upon the nature of the substrate that you're using for the production of this particular PHA molecule. So that is actually what I would like to emphasize. So extraction, since this is one of another area where I actually want you students to focus on, because we have tried, this is one I really want to tell you, like this is one of the area which I basically personally feel that till now I was not able to succeed to devise a green method for this particular one, because eventually the process which is giving a huge yield or higher yield is always with chemical, because after extracting you have to break open the cells, you have to extract this polymer and you have to precipitate it. All this requires series of extraction methods. You can follow alkaline extraction, you can follow acid extraction, you can follow detergent based extraction, you can follow solvent based extraction. So the problem with all these methods is one way or the other way, they are using some chemicals for extraction, and eventually after extraction, these chemicals are entering the environment one way or the other way, right? So one way we are trying to save the environment, and the other way, this generation of this chemical waste is actually coming to be inevitable. So this is one of the area where researchers are focusing to devise a green method or a less polluting method for the extraction of PHA, and they are still working on that particular thing. And there's a huge scope once you can find a simple methodology for the production or extraction, for the efficient extraction of the PHA from the bacterial cells. So you may have a focus on this particular thing of extraction also. So that can actually be a beneficial for all over the scientists who is working over the entire world. So this is basically what is happening in the methodology. So as you know, this is an intracellular molecule. So this is a bacterial cell, this is an intracellular molecules. So you can have any of the method of cell disruption whereby you will be recovering this PHA. But the problem is that the PHA should not be disturbed. Your selection of your extraction method should be in such a way that maximum recovery of the PHA should be there and minimum loss of PHA. Of course, since any extraction method, you cannot always recover 100% recovery cannot be achieved. There will be significant proportion of loss also. So the process should be designed in such a way that you have maximum extraction possibility. So maximum amount of PHA is being extracted. So accordingly, you have to design your extraction process. Now, how will you do the characterization? I'm not explaining too much. I'll just show you the spectrums. In a research, if you are doing an academic research, of course, for publishing your paper, you require the characterization data is very much important. Otherwise, they won't be accepting. So you can do a simple TLC and FTIR from or UV spectrum from your own lab. And XRT, I think in most of the colleges now it is available. And some of the sophisticated analysis, which you may require the contact of external this one external source is your NMR analysis, your TG analysis, and your DSC analysis. So all these things together may cost 1,000 or 1,500 only nowadays, because there's a lot of institutes which is doing this particular thing. So that would be the cost of what for the analysis or history analysis are. So just show you like how the sample looks like. So this, if you have the thin light chromatography, and there are different solvent systems which could be used. So you have this is the first test. You will generally have a RF value around this particular point, and you will generally have a brownish yellowish color spot in your TLC plates or in your TLC slides basically. And UV spectrum, the characteristics of the pH standard as this polymer what we got from Bacillus subtilis, I think. So this is 240 nanometer. It has a sharp peak at 240 nanometers. So all these things are preliminary confirmations. We cannot confirm with this. So we went to XRD to study the characteristic peaks. So PHP generally has a characteristic peak at 33.5 and 68 actually. So almost we got significant peaks on that particular one. And this is the DSC. So why we are doing DSC and TG is DSC and TG are the analysis which is done so as to understand the feasibility of thermoplastic application. Till what point this particular polymer can withstand the heat. Because plastic generally you have the melting procedure, right, for molding it into a sheet or a vessel or whatever it may be. So before it is being applied to an industry, we have to have a strong foundation regarding till what point this can withstand the heat. So to understand the thermal degradability pattern, and to understand the melting temperature, we use this particular thing, and this is how the pattern would be there. So you can compare it with the standard also if required. And this is the TGA spectrum where you have the degradation pattern. So DSC as well as the TGA is supposed to give you an idea regarding till what point the thermal degradability of the polymer is there, and accordingly you can have your application studies. If you want, you can blend it with some other. If you want a much more thermal resistance, you can blend it even with some of the biodegradable polymers, maybe cellulose, chitosan or something, and then you can improve the material property of that particular one. So that is the idea. And NMR is a wonderful technology to finally characterize the molecules, and PHA generally have three sharp points. One is at 1.2 ppm, the next one is at 2.5 ppm, and you have 5.2 ppm. So these are the characteristic peaks which is concerning the PHA. So once your extraction process is like completely paka, you will get the peaks like that. Okay, so it's easy to get the things, but only thing is that the impurities should be minimum, since NMR is a very sensitive methodology. It is easy to get the peaks also. So while you're working, you should take all the necessary precautions to avoid the impurities before you're giving that polymer for characterization. Okay, and again C13 NMR, these are the spectrum. C13 NMR, even if you're not doing also, that's okay, because most of the papers are recommending H1 NMR once you want to publish those things. So this is an outline regarding how various kind of waste could be utilized for the production of this PHA. And what about its application? Why I want to work on PHA? So the applications of PHA are actually like, I cannot put that thing even, I can even take a lecture with the application of PHA. So that will be again a topic of another lecture basically. So the first one, of course, is for the production of bioplastics, say like plates, maybe spoons, the degradable ones, the coverings, the food coverings, the food wrappers, what you're using, all those things. That is the first application. Second one is the major part and the most costly applications. Why this is required is the bio implants, like for the medicine, for drug delivery, for kind of production of various kind of materials which should be implanted inside our body. For that one. Next one, I explained you the possibility of converting this PHA into methane, and thereby it could be used as a biofuel, right? So for biofuel production, that is the next one. And recently, and now myself is also working on this particular sector. PHA, there are very few references, 3 papers for my knowledge, and one of the other paper what I have published recently is a review of literature in another Scopus journal, which is regarding the application of PHA as an immunostimulant in the field and how this immunological patterns. Basically, I have worked with fishes, but still there are high possibilities of using them as an alternative to antibiotics in the feed also, and to study the immunomodulatory activity, because these are short chain fatty acids, and short chain fatty acids are supposed to have a good immunomodulatory properties. Then endotoxin removal. I have not worked actually with this particular thing. I don't want to comment on this particular thing. There are a few papers on this one because I have, the one which I have not touched, I don't want to comment. So this is again another possibility. If you are interested, you can actually work on this particular food grade surfactants, because most of this PHA producing one will be biosurfactant producing one also, and an integrated approach could be used for the production of biosurfactants and biopolymers over here. And these are used in some of the fine chemicals and medicines. And the bioprinting, that is one of the major blooming sector where you use degradable ink. For that one also, for as a stabilizing agent, PHA is being used. So these are the very few applications what I listed over here. So as I told you, just simply browse like applications of PHA, you'll get a review paper which will be containing almost 300 to 400 different applications. So that is a demand for this particular polymer. Okay, so this is actually the thrust of what I actually wish to discuss with you. And with this, what I'm going to tell you is, see, it is sometimes sitting at college. Once I was starting my research work, probably I was starting the research work from with PHA from my MSc days. So this was started from my MSc project till this, where I have my own PhD students who are working with PHA. So still I'm working with this because the inspiration is like we still can work with the limited facilities whatever we have done. So all these things are based upon my MPhil and PhD work publications basically. So I'll show you like how we have worked. Okay, so the first one is regarding how we can produce PHA from carica papaya waste. The papaya peels were utilized for the production of PHA by using this one. It has been published in Arabian Journal of Science and Engineering, which is again a Springer publication. So this give you an outlook like it is possible. It is possible even for you to do that thing in your this one. So this is another interesting work which we have done in our in my MPhil days. So anti-candle activity of bioplastic sheets. So this PHA generally on its own will not have any antibacterial activity because it's just like a plastic. It doesn't have its own antibacterial activity, but still you can incorporate some extracts like neem extract, tulsi extract, or some other thing which is having good antibacterial activity along with this one during the plastic sheet is being formed, and thereby you can have antibacterial packaging so that shelf life of food can be increased, and multiple other methods. So this is yet another Scopus publication at that time basically. The next one is the statistical optimization. So this you require the help of a person who is well versed in optimization. There's something called as RSM optimization, response surface methodology. After your one factor at a time, you have to go to the statistical optimization. So if you are interested in pursuing a PhD on this particular thing, you can actually have this. And again, we have published this particular thing in another Springer journal, which is Arabian Journal of Science and Technology. Again, with a good impact factor. So this is one of the interesting thing where you can work and explore the possibilities. Next one, we tried off, my basic habit is this because I don't want to actually fix with one of this particular applications. I always have to have a quest to lamb to different applications. So what I thought is, why don't I apply this thing for the drug delivery purpose? So this we have actually tried it with synthesizing the nanoparticles, because that was a time where I was actually interested to study a bit of nanobiotechnology. So I just extended my work in such a way that the pH is converted into nanoparticle, and this particular thing, the whole synthesis is from orange peel based medium, and this is used for the delivery of a drug, that is which is generally used for your ophthalmic drug delivery for eye infections. And luckily it is published in your Taylor and Francis Journal, International Journal of Polymeric Biomaterials and Biomaterials. Then again, we continued our journey, we explored the other possibilities. So recently, I think for the last year, we got some of the publications from, we were actually working with distillery effluent. So we thought of utilizing that particular thing as a production. So we are just, our team were working with this particular thing, and we were able to produce that thing. Again, that is again published in another one of the reputed Taylor and Francis journal, which is Biocatalysis and Biotransformation. So this all requires a simple laboratory space and all the simple facilities, whatever is available in the college to work out with. And recently, it was a privilege to have an invitation from the most, world recognized group, that is by Professor Kudus, who is the one of the pioneer working with PHA. So they invited for submitting a kind of book chapter in a Springer book which they edited, and we have contributed. This is in 2021, so we have contributed. Myself and my team has contributed a book chapter in which I have described clearly like how fruit waste could be used as a sustainable alternative for the PHA production. So it is again a privilege, like a person from a background like what you have today is being invited by a world-renowned scientist for contributing. So this what I would like to tell you is not my greatness or not the team greatness of my team, but actually you can also do that. It's not impossible, like hearing a lecture and thinking like only with a big lab space I can do. It's not like that. You can even do that thing from your college space over here. And all that what I talk to you is all about PHA, and I told that there are general other things about what we work with. So this is it is out of curiosity, because I always think of this egg waste, because you know that we are producing a lot of eggshells per day, wherever you are, whether you are in Tamil Nadu, Kerala, Karnataka, whatever it may be. So why it could not be used as for the production strategy? Why it cannot be used as producing? It cannot be used for the production of PHA because it doesn't have lipid, it doesn't have your carbohydrate, it doesn't have protein. Majorly it is having calcium carbonate. So this was actually rolling on my head from the day I was starting my PhD. But I couldn't find an alternative how to use it. So, but after reaching here last year, we were just thinking about working with another biopolymer, which is your hydroxyapatite. So, hydroxyapatite is another biopolymer which is used for the production of your implants, basically. And our team has worked with this particular one, that eggshell biowaste for hydroxyapatite production, and after a long struggle, so the story is a huge struggle, and after a long struggle we were able to publish that thing with an environmental research which has got a impact factor of around 6.5 or so. Currently, not talking about bothering about the impact factor, you don't worry about those things, that is all secondary, but communicate your research in standard journals so that it will be beneficial for the other researchers also to take up from where you are leaving, because I may not be able to continue some of the work because of my busy schedules or because of the lack of availability, but somebody else can continue it from that point. So communication in the standard journals is the most important part of your research. It should be one part of the research. So I would also have to emphasize that you can also work on some other ways. Not only polymers, you do with enzymes, you do with any other kind of metabolites, fertilizers, everything. And but use this waste valorization concept. Okay, so rather than using synthetic medium, try to use any kind of waste for the production, so that waste utilization plus a product formation can also be done. You have a lot of entrepreneurship availability supported by Government of India for this particular thing. A lot of fund is also being allocated with this particular one. Okay, so with this, let me conclude, since I've talked majorly with the PHA. So PHA continues to be the most widely accepted alternative to synthetic counterparts. So they are fully biodegradable, non-toxic, and biocompatible. They have material properties which is closely resembling the synthetic counterparts, but the problem is cost of production and solvent extraction. So cost of production, you can utilize any of the waste. So we are almost successful in solving that issue, like utilizing a waste. But the second thing require much more focus, that is the development of a greener method for the extraction of the BHA. So I actually want students, because you have much more potential to find out an alternative to this one, so that we can actually make its commercialization important. So concluding with a remark, so it's all about saving and preserving her integrity, because nothing can convince once she responds. That is the basic lesson what we have learned in the past 2 years at least, because we have been witnessing before the corona itself, we have been witnessing a lot of natural calamities all over the world. Amazons was burning, one way Kerala and Chennai was hit by severe kind of flood. The north side was affecting with severe drought. All these calamities we were experiencing, and it's not simple natural calamities. It is actually a remark from the nature. It is actually a what to say? It's actually a warning from the nature that over with her power or in front of her power, the superhumans even cannot survive, because we have been troubling her a lot. We have been troubling her integrity a lot. So at least whatever we can do, let us do to serve her integrity, because we don't have any words and we don't have any weapon to combat ourselves once she starts responding. Okay. So with this, let me conclude my presentation by thanking one and all for giving me an opportunity for this particular session. So thank you so much for the organizers, and thank you so much for the audience for inviting me and letting me share my views, because I don't think that I am an expert on this particular area. I'm just a learner. I'm just a beginner in this particular my career as well. And so your insights, your ideas, your this one will also be helpful to me. So it will be a pleasure once to answer your doubts if I know. And if I don't know, also it will be an opportunity for me to learn from your doubts and to explore that particular thing. So thank you so much, and over to you.
Thank you, sir. Nice presentations. The topic is very interesting. Thank you, sir. Participants, if you have any doubts, kindly interact with our guest. Audience. Okay, I think no questions from audience side, sir. So once again, thank you, sir.
Thank you, ma'am.
Thank you, sir. Now I request Mrs. Violet Beulah, Assistant Professor at our department, to deliver a vote of thanks. Ma'am, please.
Good morning, ma'am. Am I audible?
Yes, ma'am, you are audible, ma'am.
Today, my words are not enough to express the gratitude. On behalf of our microbiology department, Ratnam College of Arts and Science, I would like to thank our chief guest, Dr. Mridul Umesh, Assistant Professor, Department of Life Science, Christ University, Bangalore, who graced us with this thought-provoking address and detailed explanation about biopolymer. I would like to thank our beloved principal, Dr. Murathan, for all words of wisdom that radiated a source of energy within us, and CEO and Secretary Dr. Manikam for providing encouragement, opportunities, and support. Here, heartfelt thanks goes to all our faculties. We feel proud and thank you for making us feel happy with your presence to this seminar. Last but not least, I thank all the students for showing their interest in this program. Once again, thank you very much to all who attended this 2 days national seminar. Thank you.
Thank you, ma'am. So once again, thank you all. So the platform will be available in another few minutes. The feedback will be posted in the chat box. So kindly fill your feedback and get your certificates. Thank you. Thank you all.
Thank you, ma'am. I can leave, no, ma'am.
Yes, sir. Yes, sir. You can use.
Thank you. Thank you so
Thank
much.
you.
Yeah. Thank you
Thank you,
all.
sir.